And go. Okay. Hi everybody. So we are only three on this room now. We actually just did the June seminar with 45 people that joined, so that was very nice. But we were not able to record this seminar, so we just enter a new Zoom room, and we are now recording the talk of Bárbara. So Bárbara comes from Mexico in the UNAM University, and she will present her work with sedaDNA.
Thank you for inviting me. And it's a pleasure to present my work entitled Metagenomics Analysis of Lake Chalco sediments with a large taxonomic diversity, providing insight into the Holocene of Central Mexico. I would like to start with the question that we make us for this study. What were the change in biotic diversity related to climate, environment, and human impact corresponding to the Holocene period? And to answer this question, we retrieved a 2.5 meter deep sediment with a piston core in the lake bed, in the dry lake bed of the Lake Chalco. Lake Chalco is located in the basin of Mexico and constitute one of the several lacustrine depressions along the Mexican volcanic belt. And it is also considered one of the most ancient lakes in the transition zone between Nearctic and Neotropical region. The lacustrine sediment are thick, 300 meters, and contains around 400,000 years of paleoclimate and paleoenvironment history, thus representing one of the largest records. Previous archaeological studies have indicated that human occupation in the center of Mexico were around 6,000 years before present. However, there have not been sedaDNA studies to characterize the biodiversity. Lake Chalco is one of the three lake systems that in past was just one big and deep lake. To guarantee the sterile condition, we built an in situ laboratory and implemented measures such as the bodysuit, face mask, gloves, and we sampled in the center of the cores, frozen immediately in liquid nitrogen to transport to the laboratory and store it at -80 degrees. All the DNA extraction were conducted under clear conditions following the standard protocols. Here I am showing you the experimental and analytical strategy. We sampled from this 2.5 meter deep sedimentary sequence samples for sediment smear slides to perform the fossil diatom analysis. Also for X-ray diffraction to perform the geochemical analysis. And finally, previously we took samples for metagenomics analysis. We performed the DNA extraction, the shotgun sequencing, and the bioinformatics analysis with a pre-process, and then we upload the sequence to the MGRAST server to do the taxonomic and functional classification, and also we use SEED and Metaprot database to do the functional genes annotations. After this, we performed the analysis of the results in our environment using several packages. Here I am showing the combining analysis since the lithology, the biodiversity, based on metagenomics analysis, the fossil diatom analysis, and the geochemistry. Based on fossil diatoms and geochemistry, we propose three past environmental zones, the subsaline, hyposaline, and freshwater. And we wanted to see if these metagenomics results fit with the zones proposed. We also performed the beta diversity analysis, and we wanted to see if the sample clusterings in the zones proposed, in these three zones proposed, And we, as we can see here, the taxa and protein analysis based in Bray-Curtis analysis almost did the pattern that we propose in the three zones, just in the case of the 60 centimeter samples and these 185 centimeter samples that grouped in different positions that we proposed. In the case of these 60 centimeter samples in taxa and in proteins analysis, as we can see here, grouped with the most superficial samples, as we can see here. And these 185 centimeters samples grouped with the most deeper or freshwater samples. We also performed the NMDS analysis to see how the TOC, titanium iron and manganesium iron combined with the metagenomics analysis. And we can see also the same pattern. Here are the most superficial, the subsaline samples, and this one is the 60 centimeters samples. And here in circles are the deepest, and this one in the triangle is the 185 samples and grouped closer to the deepest. And the same when we repeat the analysis just for bacteria and archaea and also to eukarya, we obtain almost the same pattern. Then we propose two transition zones, one between freshwater and hyposaline and other between hyposaline and subsaline. Here I'm showing the top 10 families reveals the abundance change over the sedimentary sequence. In bacteria is not so evident, but in archaea and eukarya, we can see the differences between layers in abundance of families. Especially in eukarya, we can see this big light blue pattern is corresponding to the poaceae, the grasses. And we notice the highly abundant of this family in the subsaline zone. We wanted to go deeper in this cyanobacteria abundant analysis because we noticed that high abundance of these cyanobacteria were mainly in hyposaline zone in this two, in this case. And we also found Synechoccus cyanobacteria highly abundant in this hyposaline zone, is the pink bar. And this cyanobacteria is related to eutrophication. And then we propose that this result and the TOC highly associated with this hyposaline zone could be suggesting an eutrophication of the lake. We also go deeper in the fungi analysis abundant, and as we can see, again, the highly abundant were found in hyposaline zone, and as we can see in these bars. But here the novedos were that we found especially genus related to mature forest in freshwater and hyposaline zone. And surprisingly, in these 6,000 years samples, we didn't found any annotation to fungi. But in the subsaline zone, the most superficial, we found several diversity and abundance of several microscopic and pathogen fungi related to crops and some insects. Then to summarize, we propose three zones: freshwater, hyposaline, and subsaline over the Holocene. The freshwater, we propose that lake was deep, cool, fresh and mesotropic with anoxic bottom water. And we found some mycorrhizal fungi. In hyposaline, we proposed that the lake was shallow, warm, high salinity levels, and eutrophic with anoxic bottom of water. Here we found the boom of cyanobacteria and also some diversity of fungi. And in this subsaline zone, we proposed that shallow temperate diminish the salinity levels and eutrophic lake. We also proposed 2 transition periods, one from freshwater to hyposaline, from a cooler environment to a highly warm one. And the other and more evidence from hyposaline and subsaline, where we found around to 6,000 years the first settlements of the humans in the center of Mexico. And we found a boom of mosquitoes here and a drastically change of the environment from a mature forest to grasses lands with some crops. And in subsaline also we found this pathogen fungi and some pathogen microbials that I am now showing in this talk. In conclusion, metagenomics result correlates with fossil diatom and geochemistry zones proposed and let us infer change in the past environmental at the Lake Chalco, and we evidence a transition state between hyposaline and subsaline, and we evidence that for the change in vegetation, for the boom of mosquitoes, especially the Culicidae family, higher precipitation related with titanium, pathogenic fungi and crops. And therefore, we propose that the first human activity had a large and rapid impact on the aquatic and terrestrial ecosystem in this area. And finally, I would like to thank to all my collaborators in this big and challenging project. And thanks to my current boss, Dr. Maria Vilas, for her support, and my lab mates. Thank you.
Thank you very much. Thank you.